And today I would like to talk to you a little bit about the research that went behind my abstract, which I've titled Detecting and Assessing the Epidemiology of Asymptomatic Malaria in Regions of Sub-Saharan Africa. Before I get into the actual meat of my presentation, I want to give you a little bit of a guide about the progression of my talk. And the reason I want to do this is I just find it a little bit easier to keep your bearings while we're quickly going through the things I want to talk about. So the first thing that I'm going to do is define the disease that I work with and talk about the data that was available to me when designing the research that follows. Then I want to spend the majority of the time talking about how I developed a detection methodology for my disease. And then finally, I'm going to talk a little bit about some of the trends of interest that we have already seen in the data and how we plan to move forward. Now, as many of you are probably more familiar with than a US audience, malaria is a very big global health concern. As you can see, the WHO reported in 2019 that there were 219 million cases that year and 405,000 deaths. Now, the reason I highlight this is because the major burden of this is actually put on the WHO Africa region, which is how I paired up the place that I'm studying, what I'm studying, with where it matters the most. So a few things about the disease itself, which are just interesting and not necessarily consequential to the research right now. But malaria is caused by a parasitic unicellular eukaryotic organism of the Plasmodium genus. And there are currently 5 species known to cause disease in humans. The 4 bolded species that you see here are of consequence to the research that I'm going to go over, mostly because of geographic bounds. So these are the only ones that we have to concern ourselves with right now. If you live in an endemic region, symptoms of malaria are fairly recognizable if they occur and include things like a cyclic fever, headache, and chills. I've highlighted this because a lot of my research deals with people who don't show symptoms. So it's really important to just assert that it's something that you can easily recognize if you've had it. So let's talk a little bit about elimination. Now, elimination has been a really big goal of the field since the 1950s. A really big thing that highlights this is actually the start of the CDC in the US, which is the Center for Disease Control, was actually brought about because we wanted to eliminate malaria here. And since then, it has been a global effort to eliminate it everywhere. One of the really big pillars of elimination currently happened in 2016 when the WHO estimated that we could reduce all local transmission by 95% by the year of 2030, which is all the way over here in this graph, which I'm going to explain in just a second. Now, in this 2016 assertion, they gave us quite a few milestones to hit so that we know we're actually on track, which brings us over here. This is from the most recent 2020 report. If we were actually hitting those milestones and on track to reach our goal by 2030, we would actually be following this green trend line right here. But as the data shows, elimination isn't occurring as quickly as we thought it should. We're actually following the slower slope of the blue line and projected to continue to follow that. Now, there is obviously a pretty big difference between this line here and then this line here. There are many different things that can contribute to the hindrance of elimination, and these are the trends I just want to note before COVID-19 interrupted a lot of the efforts for malaria elimination. So we were not on track prior to that either. As I said, there are many things that have been theorized that are contributing to why reality is not necessarily matching up with our expectations of elimination. But my lab has a particular favorite, and that is the existence of these asymptomatic individuals in all endemic populations which carry the parasite and thus can feed the parasite back to the mosquito without being detected. And that's because most of the elimination efforts, other than the widespread ones like bug nets and spraying, tend to address only the symptomatic cycle, because it relies on identifying the individuals through symptoms to treat them quickly and put them as part of the surveillance efforts. So because this is the biggest part of what's addressed by the WHO, we're completely missing this other reservoir for the parasite, which is thus hindering how quickly we can get rid of it because they're just not being shown. Which brings us to the data that is going to be utilized in this overall study, and specifically the detection development for this particular presentation. So all of the data that I use comes from the Africa 6K Top Med project, which was funded by NHLBI, which is the National Heart, Lung, and Blood Institute in the US. And it has around 6,000 individuals included from a wide range of countries from the African continent. And the reason I say this is because several of these countries are in different stages of their elimination efforts. So it gives us a good range of things that we may see as elimination progresses forward in the realm of asymptomatic individuals. So I do want to note that each of these populations also has some measure for us to actually confirm that these people are asymptomatic. For example, like temperature or self-report of no active infection in the last 2 weeks. Now, the reason that this is important, because if someone's carrying the parasite but doesn't have a temperature, this means that they're likely not experiencing the traditional symptoms of malaria and thus can be determined as asymptomatic. These individuals have many different variables collected on them that aren't just the sequencing. So they have cardiovascular health variables, general demographic information, and these will have higher consequence with following research, but right now we're going to focus on how all of these individuals have PCR-free whole genome sequencing data, which is the only thing we used for this part of the study. So you know what disease I'm working on, why it matters, and where all of the data came from. Let's now get into developing a detection methodology for malaria using only sequencing data. Before we can get into the new methodology, we need to give a little bit of acknowledgement to the old methodology and how it led me to begin to develop this new version. Now, I do want to note that there are two different ways to talk about diagnostic or detection methodology. It's those used in the clinic versus those used in research. Even though all three of these are accepted as diagnostic methodologies, generally either blood smear or rapid diagnostic tests are used in the clinic, and research is more saved for the nucleic acid detection. And the reason that I find this particular method of diagnosing interesting is because it's based on PCR amplification of a parasitic gene. It's the 18SR RNA gene. And the reason that this is interesting is because it shows us that parasitic DNA is actually in all of these individuals' DNA samples that would have been submitted for sequencing. If we can amplify up this gene using normal PCR methods, then there's no reason that the sequencer that received the same sample wouldn't be able at some point to sequence some of the parasitic gene. This sort of framework is what went into the idea that maybe we could pick up these reads that belong to something other than the humans in the sequencing process. Which brings me to the theory behind my research. Along with knowing that some of these individuals could amplify up the parasitic gene, I do want to highlight that in our sample, the Africa 6K sample, anywhere from 0.01% to 2.9% of the overall sequencing reads were not mapping to the human genome, which means that there's anywhere from 60,000 to 17.4 million non-human reads available for alternative alignment. So this, along with knowing that the parasitic genome is in there, brought me to the idea of being able to align it to the Plasmodium genome. And we started this, if you can see over here, with the mitochondrial DNA. So let's see if we can combine these two theories together and actually make this detection methodology work. Well, let's go directly into how well it did work, and then we'll kind of diagnose how we got through the entire process of making it specific. So the percent agreement, and I do want to note here, generally in the field of malaria, the gold standard is blood smear. But all of the samples that we're using here, what we're using for comparison is the PCR-based methodology that I showed you about 3 slides before, and comparing that to the new sequencing methodology. So we found that they were about in the binary call of infected to uninfected, 78.6% similar to each other. And we also found that the new methodology, the sequencing methodology, is actually better at identifying true negatives rather than true positives, because specificity is the proportion of people who were called negative are actually negative. And these two values that go here are related to the sensitivity and specificity of the test. This is positive predictive value and negative predictive value. After looking at these general terms of how well our assay is doing, we wanted to see if it was performing also in picking up the different species, as well as it was just the binary calls. So what we used to do that was the weighted kappa statistic, which is where you consider each of the different species and their combinations their own test. So then you compare it statistically. Okay, did it get it right on falciparum? Yes or no? Did it get right on vivax? Yes or no? And that comes together to be able to give us a percentage of concordance. And we found that it was actually relatively high at 84.5, excuse me. So at baseline, in the people that we tested, it wasn't the full population, just so you know, it was around 1,000 people. The sequencing methodology is actually performing quite well and showing us very exciting results. Let's start unpacking why we started with the mitochondria and how that is functioning as a viable target at this point. So, the reason we selected the mitochondrial DNA was twofold. The first one being that mitochondrial DNA is actually usually at a much higher copy number than autosomal DNA. In the parasite Plasmodium, it's lower than you would expect of a eukaryotic organism, but still reported to be about 20x over the nuclear genome by this paper. And that's just because Plasmodium tend to only have 3 or 4 mitochondria as compared to other organisms, which can have much more than that. And then also we picked the mitochondria because the mitochondrial DNA lacks the proofreading mechanism that the nuclear genome has. And thus it makes it theoretically more likely to diverge more quickly, meaning that the different species would separate in base pairs more quickly than the nuclear would because there's nothing correcting it. And yes, I know that this graphic over here is quite a lot to look at, which is why I'm going to spend the next couple slides breaking this graphic down for you. This is what's called a Circos plot, if anyone hasn't seen it. Let's start unpacking this very difficult graphic by walking from the outside inward so that we can understand what it's saying to us incrementally. So the first thing I want to know is these labels on the outside are the species of Plasmodium that specific range of nucleotides indicates. And then the _M just means that this Circos plot is specifically for the mitochondria. And then finally, I do want to note that each of the species has its own color. So anytime you see this specific color and not necessarily the lighter version of it, means that this indicates that somewhere in this analysis was, for example, Plasmodium malariae. Now, what this means is if you see here, this color is also right here. In this row right here, it means that malariae is being compared to vivax. How is it being compared? What does that mean? Well, if we walk another concentric circle in, what we actually did here was a sliding window base by base comparison using Hammond's distance, where you measure the similarity in that little 200 nucleotide region, and then walk all the way across the rest of the genome so that every 200 base pair combination can be compared for similarity. And what we found was that there was a pretty tight range of similarity for all of the Plasmodium species being set. They were 79% similar to each other, all the way up to 100% similar to each other. Now, it's really important to remember that this range is pretty tight and kind of high, because these dips and valleys that you see look like there could be quite a bit of difference between the species that is more artificially there than reality. One thing I do want to say is that the exception to this percentage difference is actually the difference between the two different species of Plasmodium ovale, which is Curtici and Walliceri. We actually found that these were not anywhere less than 97% similar between the two of them. So for the rest of the talk, anytime ovale is referenced, we have to consider these two things the same, because at this target depth, we're never going to be able to tell the difference between the two species. So all that being said, all of these are just the pairwise comparisons of each species to the other top species that it's under. Through this, we found that there is a potential informative region. Now, what this means is there is a region where the species are at least less than 92% similar to each other. And this is important because in this region, we may be able to use it as a DNA probe to tell us the difference between the species if a reed should land here. So for example, if a reed lands here that comes from Plasmodium falciparum, we should be able to tell that it is actually Plasmodium falciparum based on the differences. Finally, we actually have one more circle to move in, and that is these different ribbons that you see in the middle. And what this is a mauve multiple alignment comparison between the species. And what this does is it takes all of the different genomes and separates it into these things called LCBs. And LCBs are just generally these regions that can be considered evolutionarily the same. So what we would want to see in an ideal DNA probe would be that there are regions where these LCBs are not touching each other, because if they don't touch each other, that indicates that there's a space where the parasites are unique enough from each other that it's not evolutionarily conserved. But as you can see here, all of the species actually have touching LCBs, which means that we're working with something that is very similarly related to each other. And another thing I just want to note, in case you're looking and say, hey, there is difference because the order of the colors is not same per species. But I do want to just remind you that the mitochondrial genome is circular. So even if these chunks are not in the same 1 to 0 or 1 to 100 range, they are actually in the same order because all of the colors are the same in each of the parasites. It's just that the origin of sequencing for the reference was not necessarily the same. So this all comes together to show us that we've identified a region, even though it's in an LCB of 960 base pairs, that could potentially serve as a very viable DNA probe to be able to use to differentiate the species from each other. Which brings us to the general conclusion, and I'll tell you how we got there very generally in just a second. But what we found overall when we began to compare these mitochondrial sequencing runs by aligning them to the unmapped reads is that the mitochondria actually functions as a very good methodology to call if a person has Plasmodium, but we're still working on it to get it a little bit better at differentiating the species from each other. And the way that I did this just generally was by taking the reference genomes in that little area that I was telling you that we were interested in, and making simulated reads and seeing how well each of the species were differentiated from each other. And based on the work on that, we have, we're just looking forward to finding other ways to make it a little bit more species specific. So all of this comes together to show us that we have a way of detecting Plasmodium retrospectively using only sequence, which is really, really exciting and is very, very impactful for anyone who's working in endemic regions who may not have pulled together the resources to get blood smears on everyone they collected. So you generally know how the detection methodology works and how it came together. So it in comparison to the PCR was very good, and we found potentially informative regions to use as DNA probes. So in the end, what is the information that this will give us that excites us truly? Let's start going over where the epidemiology could fill in some gaps for us. Which brings us to the first thing we noticed as soon as we began using this detection methodology. Unfortunately, we actually think that asymptomatic individuals are a much bigger problem than originally expected. And we came to that conclusion based on one of the subsections of the Africa 6K population, which was the Ghanaians. And the reason we say this is because all of them were collected in the Brong-Ahafo region in and around the capital of Sunyani. And that region has a pretty good malaria reference to go back and look at and say, okay, in the years that it was collected, for example, in 2005, the malaria incidence was reported to be 29% in the region. But what we found using our methodology was up to 71% of the population per year was actively carrying at least one species of malaria parasite. And none of these people reported to be actively ill in the last 2 weeks. So this means that even if some of them may have been reported in this 29% of infection, there's a big portion, even if we take that 29% out, of these people just existing without being detected and reported with the malaria parasite. So this is going to be a really important implication of finding how many people in these 6,000 are actually infected with the parasite with no symptoms. Next, let's go into some of the species thing that we have seen. So when you're developing a new methodology, you always want it to make logical sense. And one of the things that really indicates to us that we're seeing exciting results is in Africa, specifically in the sub-Saharan region, the majority of infections are Plasmodium falciparum. As you can see here, this is a map taken from the Malaria Atlas Project, and it's actually from 2005, which is where most of our data that has already been analyzed came from. And as you can see from these bright yellow regions, most of the infections that are seen are Plasmodium falciparum because it has a really, really high incidence in this region. And what we found in our data of the 1,500 individuals we have already tested, 96% of the infections that we found contained or were singularly Plasmodium falciparum. And what I mean by contained is malaria is a very specific disease that can have mixed infections, not just single species infections. So this together just gives us some sort of comfort that we're seeing the things that you would expect of this region. But what we're actually also detecting is something that is currently not been published on. To date, there hasn't been any indication that Plasmodium vivax is existing in Ghana, which I have pointed out here. Now, the reason for that is because in these dark red regions you see in this map here, there is a very, very high level, and it's not labeled here, but this right here is the frequency of Duffy negative individuals. And why this has some consequence to the appearance of Plasmodium vivax infection is because for the longest time, individuals believed that Plasmodium vivax couldn't infect Duffy negative individuals because the invasion mechanism is through the Duffy binding protein and thus the Duffy receptor. So it was believed for a very long time that Duffy negative individuals couldn't get vivax infections. However, in more recent years, and kind of driven by one of my PIs, Dr. Peter Zimmerman, we have found that there is potentially an alternative pathway that Vivax is using to infect these individuals. And so if we find a Vivax infection, for example, as you can see here, we've begun to look for it. We're going to contribute to the growing body that Vivax is existing in these populations, which is really, really important because then we have to discover how it's doing it. So this is just another one of the really exciting implications that could come out of this research and already has started to. With that, I would like to thank you for listening to my talk. I understand that we're having a live Q&A session coming up in the actual occurrence of the conference, but if I don't happen to get to your question in that time, or you have something that you want to ask me before then, please reach out to me at my email, which is jmo85@case.edu. And with that, I would really just like to thank you once again. And here are my works cited. I would like to take a second to thank everyone who made this research possible, including everyone in both of my labs at Case Western Reserve University, and those who supported this research through funding. That includes the NHLBI Biodata Catalyst Fellowship Program, which I am a fellow in cohort 3, and then a T32 that I received at Case Western Reserve. So thank you very much.